I got into scaling rhythms by accident because as a young man I thought marine biology was glamorous. So I went to the University of Liverpool to do marine biology. It involved working on the Isle of Man, which is a lovely spot in the Irish Sea, but in the middle of February it's not at all glamorous. But there I was studying tidal rhythms, 12.5-hour cycles in the invertebrates, and how the phase of the tide and the phase of the moon governed their behaviour and metabolism. And so by the end of that was my PhD, I then decided I'm not going to do science, it's dreadful, it's not a future for a real person. And so I bobbied off to go and do a postgraduate teaching certificate. I then saw the light, I realised I wasn't cut out to be a teacher, and I saw an advert for a job in Nature, and it mentioned circadian rhythms instead of tidal rhythms. And I still have the conscious thought in my mind. I thought, well Michael, what's a few letters in a word? So I applied for the job. It was an advert from someone I later came to know and regard very highly, Joe Herbert, in Cambridge. And Joe's an eccentric, so he could see the logic in appointing a hairy-arsed marine biologist who'd never touched a vertebrate in his life to do a project looking at melatonin and seasonality in hamsters. So it was a complete fluke. I was in the civic library in the centre of Bolton, which was the town where I did the teaching certificate, and in my lunch hour I saw this advert in Nature. And it was about the pineal and the pituitary, and forever I was getting the two things mixed up. Why he gave me the job, I'll never know.
But those are wonderful beginnings. I think so many of us come to circadian rhythms by chance.
That's
and
good.
And then you went to Cambridge.
And then I went to Cambridge, yes. And like many people in Cambridge, I went there for a 3-year contract, and whatever it is, 27 years down the line, I'm still there, and it's a great place to be. I enjoy being there. The actual project, Joe previously had worked on ferrets. He got a big interest. His interest was how external factors affected reproduction. And there were 3 lines to work in the lab. One was his primate work, looking at how social dominance affected reproduction, because obviously it's the top dog or the top monkeys who get to breed, and the subordinates have got reduced gonadal steroid activity and what have you. He was also interested in stress, the effect that stress had on the reproductive axis, because clearly animals who are stressed don't breed so well. And his third line of investigation was the physical environment, how photoperiod and seasons, per se, could affect the reproductive status. And the idea was that if you looked at these 3 different forms of reproductive control, then what was common to them would be the master switches behind fertility, which is conceptually a good idea to work on. So I was just on one of the 3 routes that Joe was interested in. And as I say, previously he'd worked on ferrets, which have a particularly pronounced seasonality, but it takes an entire year for the thing to run full circle. When I got there, the ferrets had gone. Joe had got a new grant. He decided to move to hamsters because at the time, particularly labs in the United States were working on hamsters, and as an experimental platform and as a risk to health, it's more attractive than ferrets are. And so I started there, and the task was to establish basic paradigms as to how season and melatonin control fertility in hamsters, and then look in the hypothalamus at the neurochemical pathways and what have you that are triggered by melatonin to bring about these changes in fertility.
Can you go back and give us a brief summary of what Joe did and Herbert in ferrets?
Yeah. Joe Herbert had made, I would say this, wouldn't I? But I believe he'd made seminal contributions to how photoperiod controls fertility. What he was able to show was, first of all, it's the pineal gland and the pineal hormone melatonin, which was the mediator of the effects of long days or short days upon the reproductive axis. What he was also able to show and to argue was that melatonin itself as a compound is neither pro nor antigonadotrophic. And in fact, I would guess nowadays, 20 odd years on, that's something that's still a bit of a wrinkle in the field. You still come across people who consider melatonin to be antigonadotrophic. It's not. It has no trophic activity of itself, or gonadotrophic activity of itself. It is a messenger of time. And then how the organism uses that time message depends upon the state of that individual at the time it receives the message, and also what the species is and what its seasonal strategies are. So at a time when there really wasn't clarity about what melatonin gave to an animal, Joe, in my opinion, brought that clarity to the field.
And together with Klaus Hoffmann, I think he was the second person who did.
Absolutely. So Klaus Hoffmann working on Siberian or Jungarian hamsters, Joe working on ferrets, yes. But the two of them, I'm sure, well, let me say, I only met Klaus Hoffmann a couple of times early in my career, but it seemed that they both had the same mindset, that melatonin is a time signal. It is not, per se, a pro or antigonadotrophic factor.
So they were very perspicacious and ahead of their time.
I would say so, yeah. And it was an exciting time. It was an exciting time to be in melatonin because the concepts were coming out. And then, of course, Joe and, in fact, Klaus Hoffmann had shown how the length of the night determined the length of, in their case, the activity of the enzyme that synthesizes melatonin. And then later on in time, it's possible to show that, in fact, the length of the night determines how long melatonin itself is produced by the pineal gland. And so the idea of a durational signal, melatonin is an internal egg timer, if you like, that reflects the length of the night, started to grow. It started to grow. The field, in my opinion, again, took a few setbacks because the early assays for melatonin, particularly used by some of the US labs, were not able to show this so-called durational effect of night length on the melatonin profile. So for a long time, the field went round in circles talking about whether there was a duration effect, which is ultimately what it came out to be, or whether it's a phase of effect, that the time of the day at which melatonin is delivered was the important thing. In the end, the duration model came out tops. But for 3 or 4 years, the argument went to and fro. So an important paper for me that came out of Bruce Goldman's lab. So he really capitalised on, in my opinion, on Klaus Hoffmann's observations and Joe's ideas as well about the durational importance of the melatonin signal. Long nights, long melatonin signal, it's winter. In the case of hamsters, turn off your reproductive axis. And what Bruce Goldman did was in fact to take away the pineal from hamsters, so they had no clue of season, and then using a very simple cannulation system, he was able to infuse back as a subcutaneous infusion melatonin into these animals. And of course, different animals received infusions that lasted for longer or shorter durations. And what he was able to show in Siberian hamsters was it was duration. A long signal was read as winter, regardless of whether you gave it to the animal when the lights were on or the lights were off. So there was no real phase dependence. It's not as if the animal was primed to receive a melatonin signal only at night and not at day. All the brain really cared about was how long that melatonin signal was. And great piece of work. Looking back now, you can see the line of thought that led to that piece of work. For our part, we just imitated it. We did the same types of experiments in Syrian hamsters. We'd done some assays of pineal melatonin levels, which in fact broke through the circularity of the argument as to whether in Syrian hamsters there was a durational effect on the endogenous production of melatonin. We were able to show, in fact, yes, what happened in a Syrian hamster was the same as what happened in sheep and in Siberians. So a commonality of mechanism, started to emerge. And backing up that commonality, we were able to show, just as Bruce Goldman had, that if you infuse melatonin into Syrian hamsters, you could make them think it's winter, summer, or something in between, just as a function of how long each day you give the melatonin signal. And then in parallel to that, and again addressing this pro-antigonadotrophic action, Fred Karsh at Michigan, Ann Arbor, working with sheep, was able to do essentially the same experiments. That if you give sheep long or short melatonin signals, you can convince them whether it's winter or summer. But an important observation there is that sheep become reproductively active with long melatonin signals because they come into season in autumn. Whereas, of course, in hamsters, long melatonin signals turn off the reproductive axis. So that really broke the link altogether between a pro or antigonadotrophic action of melatonin. It really was a temporal signal. It's a calendar, an internal endocrine calendar. We started to become interested in the actual, if that's a phenomenon, this durational phenomenon. Then the obvious question is, what are the mechanisms, what are the sites of action in the brain? And I remember going to a European pineal meeting where Helena Ilnerova, who had a laboratory in Prague, and she'd done seminal early work on the physiology of the pineal in relation to day length. In fact, she showed to my eyes for the first time, although Steve Ruppert and Dave Weaver in the US were doing the same experiments in parallel, by using iodinated melatonin, they were able to reveal where in the brain melatonin receptors were expressed. And it was a fundamental observation that in rats they showed that receptors were expressed in the suprachiasmatic nucleus, but more importantly from a seasonal viewpoint, they were able to show that melatonin receptors were expressed what they initially thought of was the median eminence, the back part of the hypothalamus where the releasing factors are secreted. But in fact later on it became obvious that it was in fact the pars tuberellus of the pituitary. Part of the pituitary that none of us has really paid attention to or indeed heard of until, wow, it's chock full of melatonin receptors. And following on from that really important observation, that gave real credence to the melatonin field, because up until then, you talk to anyone outside the melatonin field, pharmacologists or biochemists, whatever, say, well, what about the receptor? Well, we don't have a receptor. So you go and stand in the corner of the room. Now there was a receptor. Of course, that made so many things possible. First of all, it made possible, the galloping search to clone the genes that encoded the receptor, because now there was a biochemical correlate, so you could do your cloning experiments by one means or another, and you knew what endpoint you were looking for. It also made it possible to scan across the brain and find out which parts of the brain in this species or that species express melatonin receptors, therefore which parts of the brain may be part of the seasonal, reproductive or indeed metabolic axis. So that one, again, I can see myself back at that European meeting, seeing these autoradiographs. In fact, I think Eugene Vanacek gave the talk, but the melatonin field changed completely as a consequence of that. In fact, at that meeting, I met a chap called Peter Morgan, who'd been appointed at a research unit in Aberdeen called the Rowett Institute, with the task of, that was basically a nutrition metabolism unit, but because of the seasonal actions on metabolism, they decided to pursue seasonality as a general physiological mechanism. And so he was tasked, as they say, with looking to find out sites and mechanisms of action of melatonin. And Peter followed on from the observations from Helena of the pars tuberellus as a site rich in melatonin receptors. So he established cell culture techniques with them and was able to explore the signal transduction pathways whereby melatonin affects the signaling effects, the signaling pathways in the pars tuberellus. Beautiful biochemical pharmacological characterization of the receptor. Working in parallel, Steve Ruppert and Dave Weaver were doing similar things, not so much in the sheep PT, which was Peter's thing, but in rodent brains. And in fact, Steve Ruppert scooped the pool by cloning the genes encoding melatonin receptors, particularly in melatonin, he calls it melatonin 1A, as I always remember it to be the melatonin 1A receptor, which in mammals seems to be the one that's important for the actions of melatonin. So, again, this is another step change in the field. We got the receptor identified by autoradiography that we'd always wanted, and now we were really grown up because we had the gene encoding that receptor and a couple of related receptors as well. What's interesting, actually, and I think it's fair to say this, after the excitement of getting the genes, I think it's fair to say, it's not actually helped us really understand how melatonin works in a signal transduction, going from a daily signal, how long is the night, into the seasonal state of the animal. Having said that, of course, we're here at an SLBR meeting, and I've just been to the seasonal session. And all of a sudden, I think a new step change again has happened in this field with the recognition that thyroid hormone signaling into the medial basal hypothalamus, controlled by melatonin responsive tissues in the pars tuberellus, really could be the next move into how we understand seasonality as a mechanistic process. So the melatonin receptor enabled us to characterize melatonin responsive tissues in great detail and to locate them, but now we've had to move beyond the melatonin receptor and to think about what is it that's coming out of these tissues, particularly the pars tuberales, to control what the hypothalamus does and thereby control fertility and metabolism. We worked alongside Peter for quite a while. I had a PhD student called David Hazel-Rigg, and we got a postdoc. He worked as a joint postdoc between myself and Peter. And Liz Maywood working with me in our lab. We started, in retrospect, it might sound crude, but in fact it's the same as doing gene knockouts. We did lesion experiments. We looked at the sites of the brain that had melatonin receptors and asked the question, well, if we knock out this part of the brain or that part of the brain and give an appropriate melatonin signal by infusion back into these animals, can they read that signal or can they not? And from our work, we were able to conclude, in seroinhibitors at least, that the suprachiasmatic nucleus is not necessary for reading the melatonin signal. And to my mind, that makes a certain logical satisfaction, because the suprachiasmatic nucleus tells the pineal what to do. It tells the pineal when to produce melatonin and for how long. So why on earth should melatonin come back and have anything to say to the SCN in that seasonal context? What we also showed, which is more of a positive signal, was that if we lesioned melatonin responsive sites in the dorsal hypothalamus, then animals with such an ablation couldn't show the reproductive responses to melatonin signals, but they did show the responses in terms of their prolactin secretion. So they could read the melatonin signal for the prolactin response, which goes down in winter, but they couldn't read the melatonin signal for the reproductive response. So these animals remain reproductively active, which is a summer condition, but had low prolactin, which is a winter condition. What's personally, a bit of personal vanity now, but hopefully what's going to work out is what I mentioned previously about the thyroid hormone signaling back into the hypothalamus from the pituitary, pars tuberalis, that axis. I'm hoping that in fact they're going to link up these two observations together. And in fact the melatonin responsive sites in the dorsal hypothalamus in some way contribute to the seasonal change in thyroid hormone signaling that underlies seasonal fertility. But that's an aside. We'll see if that happens or not. Having worked on seasonality and made some contribution to the field, but not as major as we might have liked, I played football with a guy, soccer, right, with a guy, Dave Hancock, and he worked in a cancer lab, and he had made antibodies to this immediate early gene product called CFOS. And around the Cambridge area, this was becoming quite a thing. The recognition that acute stimuli in the brain could turn on genes and it could track the activity of nervous pathways in the brain by responses to immediate early genes. And I'd seen a talk by Ben Drusak, in fact, showing through mRNA expression that light pulses induced CFOS in the SCN. And I'm not embarrassed to say, I thought, well, that's great. I want some of this action. This looks really exciting. And it just so happened that the right back in our football team was an expert in raising antisera to CFOS. So we got these antisera from Dave, and we started doing immunocytochemistry. And lo and behold, as Benj showed in his initial publications, light pulses induce this immediate early gene in the SCN. And the ability of light to do that correlated with whether or not the animal's behavioural rhythms are shifted. Smart. Again, another step change. A step change in how, I thought about circadian rhythms and certainly how the field, could develop. And so there was then a sort of frantic chase and it drew us into signal transduction issues. It drew us into transcriptional issues. How are genes regulated? Well, I had to learn all this. I had to talk to Dave over the phone. Dave, how many FOS genes are there? What's the promoter sequence? What is it? How does it work? It was a very exciting time. Had to learn a lot. And ultimately, actually, it didn't pay off because FOS, I guess we accept now, is not part of the clock mechanism. At the time, there were great hopes that it may be. And again, my personal take on it is that the fly people were starting, obviously, cloning of period, and they were getting their proper clock genes. And the best thing that the mammalian people could hold up to this was CFOS, so we offered what we could. Perhaps it's a clock gene, perhaps it's not, but that's as well as the field could produce. But I think longer term, it certainly drew me, and I guess other people in the field, into thinking about transcriptional mechanisms and how the control of gene expression in general could be part of the clockwork, even if CFOS, EGR, FOSB, all the other things we looked at the time didn't turn out to be, or as far as we know, currently are not part of the clockwork. We should never say never. Then I guess the next step change was I went to a Gordon Research Conference meeting. So at the meeting, Joe Takashi's lab, terrific, outstanding, world-changing piece of work. Their production generation of the clock mutation in mice, and then beyond that, the subsequent cloning, identification by bacterial artificial chromosome rescue of the mutation, all in a pre-genome age environment. It's a stellar piece of work for the clock gene. At the same meeting, Hitoshi Okamura stood up as a hot topic and described his work with Hajime Tei and other people on the mouse period gene. So this looked to be the mouse equivalent of the period gene that the fly guys had got, and that they were showing pretty comprehensively was part of the clockwork. And Okamura stood up, gave this very brief talk, and again, it was, to my mind, it's a complete watershed, total watershed. And I remember going home on the aeroplane thinking, I'm going to give up science. I can't cope with this. I can't cope. I can't compete with this. We've got Joe Takashi doing that. We've got Hitoshi Okamura doing this. They're taking these leaps in the field. And what am I? I'm a pedestrian compared to this. I'll go and do something else. So although it excited me on a personal level, it depressed me. But anyway, once I was back in Cambridge, soon got over that, stopped feeling sorry for myself, and just got enthused in the idea that we really were, as a field, about to embark on opening this black box. And by that point, I was so frustrated that the clock was a black box, and people from other fields asked you to make interesting, insightful statements about it, and all you could say was, I don't know. Oh yes, you're working on the base of cancer. Oh, how interesting. And what do you work on? Oh, I work on circadian rhythms. Oh really? And what are they? And what do they do? And just no comparison. We just didn't have the equivalent statements to make in circadian biology that these other people had in other fields. And then all of a sudden, we now had it. It was stellar. Stellar, very exciting time. I still think we're on a high from that. I really do think, on a personal level and as a field, it really pushed everything forwards massively. And then it became frantic, and everyone looking for the how many per genes are there? If these are negative factors, as in the fly, what are the positive factors? Okay, how many positive factors can we find partners for them? And a whole range of people. Some like Steve Reppert and Joe, who'd been in the field for years, made this transition with enormous success. Other people were drawn into the field who didn't have a particularly circadian background, but obviously had the expertise and insight to take circadian rhythms as a phenomenon and then apply their expertise to sort of dissect out the mechanisms. So, brilliant. And one thing, I guess, speaking as an old fart scientist, as a young person, you might not think that things really do change, or they're glacial. Things are glacial in the way in which they change. In fact, I've been in science long enough now to see that things do change, that people do and can make a difference, and those differences are important. And it's quite exciting. It really is exciting to recognise that. Okay, you can read the textbooks and do a timeline, and Joe Bloggs did this and Jane Smith did that, and blah, 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 blah. But when you're the grad student or the postdoc, you say, I'm just pushing against a brick wall here. Things don't change. What's the point of doing all this? But in fact, if you stick at it, all of a sudden you can look over your shoulder, look back and say, goodness, how this has developed and how it's developed for the good.